First of all, thank you very much for the invitation to come here and talk about fibers. Fibers in layers diets and also some of the properties and nutritional functions. I should say that I have a bad voice today. I had a cold here over the weekend. I thought that it would have been ended now, but unfortunately that's not the case. So in case I need to retreat and take some water, you might have my excuse. Okay, but the point which I'm going to address here in my presentation, that is just some, it's not changed. Okay, that is some introductory remarks concerning fibers and also the analysis of fibers. Diet for layers and the main cell wall polymers. And then I'm going to address that the cell walls polymers, they are not evenly distributed across the tissues in the feet. Functional properties of fibers, and then also some few words about exogenous enzymes and also possibilities and limitations, and then take-home measures. So this is the point which I'm going to address here for the next 45 minutes. Just briefly, well, who I am. It may not be all who knows me, but I have been working for more than 30 years on carbohydrates and cell wall associated phytochemicals. I worked on it from an analytical perspectives, working on different methodologies to analyze the fibers components, and also looking on the nutritional functional properties, particularly in monogastric species. But if you are looking on my CV, it actually covers all the major animal species, including ourselves, the human subject. But I haven't had any publications on with layers, so why I'm here, well, you may ask the organizers. Well, few words about the fibers. Well, if you're looking into definitions, and there are several definitions on fibers, but just a sum up of what I'm considered as the fiber fractions, that is, they represent the carbohydrates and lignins that is not digested by the endogenous enzymes, either those which are secreted to the gastrointestinal tract of monogastric species or present on the epithelials of non-ruminant species. But I should also say that along with the endogenous enzymes, and if we are looking here on chickens and also on pigs, you'll see all the different enzymes that's secreted to the gastrointestinal tract, which come from the body, which are secreted into the gastrointestinal tract. In addition, we will also have different microorganisms present there, which actually, along with the endogenous enzymes, also will have an impact on the different fibers components. But basically, when we're talking about the fibers, we're talking about the part of the feed which cannot be handled by our endogenous enzymes, but by the help of the microorganisms, we will be handled a part of the fibers components. And in addition, it's also so that when you add some of the exogenous enzymes to the feed, you will, rather than using the microorganisms to reduce or to cleave down some of the bondings in the fibers components, you will have an exogenous help within the upper part of the gastrointestinal tract to reduce the size of the molecules and also to break down some of the structures to oligomers and monomers and so on. So this is within this area where we play, and it's for sure that if you're looking on the gastrointestinal tract of a pig as compared to broiler chickens, we have more microorganisms in the pig than is the case with broiler chickens and so on. Well, when I'm looking into to the fibers, and this is the methodologies we have been using for or built up over several years to make a complete characterization of the carbohydrate fractions and also the lignin content of different feedstock materials. It's so that we can analyze for the low molecular weight carbohydrates, divide them into the sugars and also the oligosaccharides. The polysaccharides can be divided into the starch, resistant starch, fructans, and then the non-starch polysaccharides and also the lignins. This is methodologies which we have been working with for quite some years, and where we also have quite a bit of table values about different feedstock materials. And for those of you who are interested in also some of the scientific readings, there is a book coming out sometime next year where we have been using or writing a paper where we have been actually showing some or presenting some of the data based on this methodology. I should also say, though, that the fiber definition has actually developed over time. Back in time, it was so that the fibers, they primarily included what was here the non-starch polysaccharides, the non-cellulosic polysaccharides, and the lignins. But today we are also considering, for instance, resistant starch and also some of the oligosaccharides as part of the fiber fractions. I should, though, say that with my presentation today, I'll primarily base it on the fibers part of it, which this part which principally will analyze the cell wall associated polymers. If you're looking into the typical diet for layers, you'll see that just roughly, this could be something what you feed your layers with. You'll see that you have the cereal part of it, and then you have the protein-rich parts. And I could say this is a diet that was used in Denmark. And when you then are calculating on the proportion of the between the cereals and also the protein-rich materials, it's approximately so that 54% that comes from cereals, whereas the remaining 46% that comes from the protein-rich materials. That ratio can naturally vary depending on your raw material composition. For instance, if you have more corn in the diet, you will have relatively less fibers coming from the cereals, whereas if you also use other protein sources, it may be relatively more. So, but that's about the distribution we have. That also means that when we are looking on we have this major part of, as for instance here, barley and wheat in the raw form here, and then we have the soybean and rapeseed meals. But one of the differences, which we also to some extent need to keep in mind, that is, while these are the raw material for the processing in the feed mills, what we are putting into the feed mills of the protein-rich materials, they are typical of some of the byproduct or co-products from the oil extraction industries, meaning that many of the protein-rich materials, they have already been processed before they enter the feed mill. Just briefly about what do we have for fibers components when we are looking on cereals as opposed to the protein-rich materials. Well, you'll see that when you're looking into the cereals, you'll see that you have Arabinose Islands, cellulose, beta-glucans, lignin, and then there's also low amounts of other components, primarily some phenolic compounds. When you're looking into the protein-rich crops, you'll see that the picture is somewhat different. You will have cellulose as the main component. Then you also have pectin polysaccharides, arabinans, galactans, siloglucans, and acidic silans, and then also lignins. But lignins in many of the protein-rich materials, they will be relatively low in many of the cell walls. The only exceptions which I really know of, that is, for instance, sunflower and rapeseed meal in cakes. Just briefly about the main cereal polysaccharides, you'll see that we have the cellulose here, and then we also have mixed-linked beta-glucans, and then we have Arabinose Islands. And when you go from and looking on the cellulose and also mixed-linked beta-glucans, you'll see in both cases they are built up of glucose units, whereas when you come to the Arabinose Islands, it's a more complex structure you have here. And in addition, it's also so that with the Arabinose Islands, there is a number of cross-linkages between the different polysaccharides. I'll now briefly go through the individual structures of it. When you're looking on the cellulose, as I mentioned before, it is built of glucose units. And it's actually the same units as also are included in starch. But in contrast to starch, it's so that the linkages between the glucose units, that's a beta linkages, whereas in starch, it's an alpha linkages. The alpha linkages can be handled by the carbohydrates secreted by our intestinal tract, whereas that's not the possibilities with the cellulose. In addition, it's also so that cellulose, the different strains of cellulose, they run antiparallels to each other, which actually make them, it's possible for them to create very, very strong structures within the cell walls. And you can have, you can view them as a form of some of the wires you have or some of the fabrics you have in a basket where shopping baskets. They give the structures, and because of the antiparallel linkage of them, or the antiparallel runs, they are giving rise to structures that is very, very insoluble and also very, very difficult really to degrade in many aspects. Beta-glucans, that is also a glucose polymer. And it's also built of beta linkages between glucose units. But in contrast to what you see with the cellulose, it's so that in approximately 25% of the cases, the linkages does not go by 1 to 4 linkages, but you have in 25% of the cases a beta 1,3 linkages. This actually has changed the structure, or not the structure as such, but it's changed a lot with the properties, while it's so that the cellulose, that is more or less completely insoluble. A very significant part of the beta-glucans in cereals, that is soluble. And the reason for that is actually that we have this beta 1 to 3 linkages, which make it easier which completely change the properties of the polymer. And then to the Arabinose Islands. The Arabinose Islands, that is the main cell wall components of the cereals. And basically it has a backbone of xylose units, and then also with side group of arabinose. And in some cases, the arabinose can be either with one or more position on one of the backbones of the xylose units here. While you could see with beta-glucans and also with cellulose that was polymers of glucose and no side grooves on it. You'll see in this case here we had a number of side grooves, and depending on the structure of the side grooves, and also in addition it's also so that there can be phenolic compounds that can be cross-linked to the xylose structures, which will influence also the properties of these polysaccharides. I've shown a picture here which I'll come back to. This is the outer cup of a cereal, where you'll see you have Arabinose Islands in this cell layers, in the aileron, and also in the endosperm, but the structure, that is a significant difference between the different tissues. And as I mentioned, Arabinose Island, that has a quite complex structures, and it also, for instance, this is from cereals, this is from the endosperm of wheat, whereas when you look on the outer tissues of wheat also, you'll see that the structure is somewhat different. So that has a great impact on their properties, and also the capability for it to be handled by the exogenous enzymes. Another component which essentially is not a carbohydrate, that is lignin. Lignin is a polymer that is formed of two alcohol units here, but the specific the special properties of the lignin, that is that they can link with either ether or with ester linkages to the polysaccharides, and also have a significant impact on the physical properties of the cell wall materials. You will see lignins present, particularly in the outer parts of the grain, for instance, of cereals, whereas you will not find lignins when you come to the endosperm, and actually not to the aileron cells either. So this structure here, that is what, and the main purpose of the lignin, that is to cement the different polysaccharides together and give it a very, very rigid structure. This was about the main components we see in cereals. In addition, if we are looking on the main NSPs that's present in protein-rich feedstuffs, you'll see cellulose, as I have mentioned already with the cereals. It's also so that you have xyloglucans, which essentially has a glucose unit here, and then also, you have a glucan unit here in the backbone, and then with side groups as xyloses. And then you have a very, very complex pectin structures, which are present in many of the cell walls, particularly in protein-rich materials. They have a different structures, whether they are present in the cotyledon or the germ, as relative to if they are present in the outer parts of the grain. The main pectin containing materials, that is the homogalacturonans. And that is also the components which we typically will hear about when we are thinking about the pectins. Pectins as a gelling agent, for instance, when you are producing marmalade. This is a pure polysaccharides that has been extracted out from pectin-rich residues. But you can also see when you look into it that the structure as such of the whole pectin complex, that is indeed very, very complex. And for instance, if we are looking again into the to the cell wall structure of pectin-rich cell walls, you'll see you have the cellulose fibers present here, and then you have the xyloglucans that is interlinked between the cellulose units, and then in addition you have the pectin-containing materials. And out of this structure, it's so that pectin in itself is the one that seems to be easiest to degrade relative to the other polysaccharide components that's present in the protein-rich feedstuffs. How does it then look when you're comparing the composition of cereals relative to protein-rich materials? here exemplified by soybean meal and rapeseed cake. Well, first of all, when we are comparing the cellulose content of wheat, barley, relative to soybean meal and rapeseed cakes, you'll see that they are consistently higher in the in the protein-rich materials, primarily because it's present in most of the cell walls that's in the soybean meal and rapeseeds. In addition, when you are looking on the sugar monomers, you will also see that the sugar monomers on the non-cellulosic polysaccharide, that is somewhat different here when you're looking on the soybean meals relative to the wheat and to the barley. The components we see here in terms of rhamnose, galactose, and also uronic acids, they are markers of the pectins in the materials in the soybeans and the rapeseeds. And what you can see that is that, for instance, with uronic acids, which are the backbone of the pectin polysaccharides, that you'll see that there is far more here with the soybean meals and the rapeseeds as compared to what you have with the cereals. On the other hand, when you come to the cereals, you'll see that it has a relatively higher content of xylose, which is actually the marker of the Arabinose Islands in cereal products, and also with regards to glucose, which is a marker for the beta-glucans. So just by looking on the sugar monomer composition, you'll notice that there is big differences between what we have in the cereals as compared to the protein-rich materials. What also can be noticed here, that is that particularly for the rapeseed cake here, you'll see that the lignin content is very, very high. And that is due to the fact that the lignin, the rapeseed, they have a very lignified hull layer, which contains very, very high levels of lignins. This was something about what actually makes up the fibers. That is some of the polysaccharides that give the structures to the feed, and also the polysaccharides and other polymers. That is some of the restrictions for the way by which the animals will be able to handle the feed within the gastrointestinal tract. But when we also, another aspect which is also important, that is that although we have the different polysaccharides in the different cell tissues, they are not evenly distributed around the different tissues. And as an example, I can take the wheat grain, and actually also rye if you like. And basically it's so that you'll see that the cell tissues that's at the outer part of the grain, they are primarily consist of 6 cell walls that is hydrophobic and essentially formed by cellulose and also silanes, and also with a significant amount of lignin. In contrast, when you're looking on the cell walls in the endosperm, which makes up this part, and you can say you have the alveolar cells which are somewhat in between, these are these cell walls are relatively thin, and they are hydrophilic, and they essentially are formed of Arabinose Islands and also mixed-linked beta-glucans. So when we come to the cereals, the Arabinose Islands and also the beta, or at least the beta-glucan, that is primarily found here in the endosperm and the alveolar cells, whereas the Arabinose Islands, they are present in some part in the endosperm, in other parts in the outer part of the grain. The structure of them are different, and so are their properties as well. And you can try to view it a little bit when you're looking on some of the cross sections of the cereals, this is from corn, this is from wheat, this is from barley, and this is from oats. And here I have only the cell tissues from the pericarp tester, which are the outer parts here, and also the alveolar cells, and then the endosperm. And looking into the cell tissues, you can see that by the way it's organized, the capability for different enzymes to work on a layer like this is very different from when it's working on a layer like the endosperm. One of the layers which are of really great interest when we come to the capability of exogenous enzymes to handle, that is the alveolar layer here. Why that? Well, in the alveolar layer, it's so that in addition to the cell walls which surround the cell content, this cell layer also contains a very, very high levels, or has a high concentration of protein and essential amino acids, together with the majority of the phytic acids which are actually present within the alveolar cell layer. If you're looking on amino acid composition of alveolar cells relative to the endosperm, you will see that particularly the proportion of lysine is substantially much higher than what you see of the endosperm. In contrast, these layers out here, they are generally very, very low in essential amino acids because it's secondary tissues, and which also are tissues. You can just more or less, when looking on it, you can imagine that these cell tissues are very, very difficult to handle. And here's just a try of an overview to look on how is it actually built in terms of lignin and cellulose and the different polysaccharides across the different cell tissues, and you'll see that it really varies. The message with this figure here is actually to say, well, although we can have about cellulose, beta-glucans, and Arabinose Islands present in the different cell tissues, the way by which the cell walls they are organized, they have a substantial impact on how nutrient can be made available. And just to look on data, which are data from fractions that has been prepared from wheat, and which are, if not completely pure, then at least rather, well, to 95% pure in terms of the different cell tissues. And you can, when you're looking down along the line here, you'll see that with regards to the different carbohydrate components, and also when you're looking on the Arabinose Islands content and the ratio between arabinose and cellulose, you'll see that the different structures, they are actually quite different. Now going to the fibers in the legumes and the protein-rich feedstuffs. I must say, compared to the cereals, the picture we, or at least I have, on the protein-rich feedstuffs, they are not quite as clear as what we see with the cereals. But we have about the same relative distribution of polysaccharides in different tissues, meaning that when you're looking on the outer part of the grain, you'll see that they are typically rich in cellulose, and they are also rich in acidic in some acidic pectins. Whereas when you come to the cotyledon or the germ, they are somewhat more they are somewhat more differently composed. Another thing which are important, that is that in most of the protein-rich materials, or in soybeans I can sell, for instance, there is no lignin presence of significant amounts. Whereas when you come to rapeseed meals and sunflowers, you'll see that you have relatively high levels of lignin present. And again, looking on a cross-section of, this is from soybeans, you'll see the same type of organization here, where we have the hull layers out here, and then we have the cotyledon down. In contrast to what we see with cereals, it's so that that in soybeans that has been processed, that means we are taking out the lipids from the grain. This has been processed, so a lot of the original structure here in the cotyledon, they have already been partly physically treated. But again, looking on the composition, the relative composition of the cotyledon relative to the hull, you'll see that there is also significantly differences in the composition of the cell walls. And the same is actually when you come to rapeseeds. And as you see here with particularly the rapeseeds, you will see that the hulls, that is very, very high in lignin as compared to what you see here with the hull from soybeans, where lignin hardly are present. With regard to the hulls from the rapeseeds, it's also so that this is a little one of the special aspects with the rapeseed. That is that in rapeseeds there is a relatively high proportion of the grains, protein, and amino acids associated to this layer. While it's so that when you are looking on cereals, you will typically see that protein amino acids, they are down to a few, between 5 to 7% of the total protein and amino acids in the whole grain that's placed in or in the pericarp tester and the hull layers. When you go to the rapeseeds, it's up to around 25%. So that is a big difference of rapeseeds as compared to many other. With soybeans, it's not the same pictures because it's not clearly as lignified as you see here with the rapeseeds. And for instance, last year I was at a meeting in the US and I saw that Dow, they were launching new varieties of rapeseeds where they have managed to breed for thinner hulls. And with those varieties, they also claim that they were able to increase the digestibility of the protein and also the dry matter. So that is some special about the rapeseeds. Now to maybe what is most important. This was it tough? A little tough of chemistry and so on. But when we come to the physical properties, because why are we interested in that, and why are we well, we are first and foremost interested in some of the chemical structures and also how it's organized within the different cell walls materials, because it can give us some of a clue on how the different types of feedstuffs, they are behaving when they are passing along the length of the gastrointestinal tract. And here I have tried to summarize some of the features. Well, as I mentioned before, the fibers, they represent the part of the carbohydrates and lignins which cannot be digested by our endogenous enzymes. And some of the properties that is associated to it, that is also that, and I've tried to illustrate that in already earlier, that is that they can protect cell contents against digestion by endogenous enzymes. And this was already known many years ago. At the time when I was a student, Saunders in California, and he actually published a paper here where he showed that the aileron cells, he was able in broiler chicken to collect intact aileron cells in the excreta. And why is that the case? Well, the aileron cells, they are built of the fiber matrix, primarily as Arabinose Islands, and also with some cross-linkages to phenolic compounds. The whole complex is very, very rigid, which makes it impossible for the endogenous enzyme to get into the cell content. And that is a way by which we are not utilizing the nutrients in the feed in an optimal way, because these cell walls, they are actually protecting the cell contents against being digested, meaning that there's a lot of the protein and also amino acids which are locked within the cell structure, which are just passing down to the lower part of the gastrointestinal tract. There, I believe, even with broiler, at least some part of it can be handled by the microorganism. So that is one of the important aspects about the fibers. Another aspect, that is also that when the fibers, and if you remember back on some of the structures you have there, they are exposed to the liquids which will be secreted into our gastrointestinal tract. It will be so that the cell walls that's situated within the cell wall, during that process, the cell wall, they will swell. And it will swell in a three-dimensional way, meaning that when you have this as the cell walls we have in the feet, when it is exposed to the liquid within the gastrointestinal tract, it will start to swell in the three-dimensional room, and part of the polysaccharides, which are making up the cell structure, they will start to leak out into the solution. And the part of polysaccharides that's leaking out into the liquid phase, that is also what are creating the viscosity of the digester materials, because that is the polysaccharides that's sitting in here and coming out into the solution. And very much depending on which type of polysaccharides that is building up the cell wall structures, they will have a different impact on the viscosity of the digester materials. And you can say that the effects of the polysaccharides leaking out into the liquid phase, that will depend on the size. That means how big the molecules they are, and also how much of it that's leaking out. And if you, for instance, are looking on Arabinose Islands and also beta-glucans present in wheat or in cereals, you'll see that basically both polysaccharides, they can leak out from the cell wall structure, and they can create viscosity. Basically, it's so that beta-glucans, they are a bigger molecule. That is the case with the Arabinose Islands, and they also have they should in principle be able to influence the viscosity to a larger extent than is the case with the Arabinose Islands. But it's also so that beta-glucans, it's more sensitive to different factors, so that means that it's somewhat easier to degrade than is the case with the Arabinose Islands. So under practical conditions, it's so that Arabinose Islands in general have a greater impact on the luminal viscosity than the beta-glucans. Another aspect that plays a role, that is the cross-linkages between Arabinose Islands. And if you're looking on the solubility of the Arabinose Islands in corn, barley, wheat, and rye, you'll see that it's increasing from corn to rye. And the reason for that, for the low solubility here in the case of corn, that is primarily that it has a very, very high cross-linkages between the different polysaccharides. And it's further so that when you're looking a little bit further down to that, you'll see that it's particularly diphenolic compounds that causes that. And for instance, when you take corn and are looking on the proportion between dehydroxyferrolic acids relative to xylose units, you'll see that it's much, much higher in corn or maize relative to wheat, for instance. And that is probably the reason to why the Arabinose Islands you are present in corn, that is very, very insoluble, and maybe also the reason to why you, in many cases, when you are using cereal for broiler chickens, you will prefer corn over some of the other cereals, simply because it stays more insoluble during passage of the gastrointestinal tract. Another aspect which is also quite important to look into here, that is because when you come to pectin polysaccharides, they are also what we often are thinking about as something that can create viscosity. And that is also true if it's so that we are working with isolated pectins. But it's not the case if it's so that we are providing the pectins in natural feedstuff materials. Under those conditions, it will give a swelling and it will have a higher water binding capacity, but it will actually not influence the viscosity to any larger extent, in contrast to the solubility of beta-glucans beta-glucans in Arabinose Islands, which will have a major impact on viscosity in the small intestine. As I mentioned before, we can have the effects of the encapsulation. I showed you a picture about a study that was performed already in the late 60s and published at that time. This is another example of data from, this is from pigs, and it's not from other layer hens or from broilers, but it illustrates quite well what the encapsulation can do with regards to the digestibility of the protein and also the fats or some of the other nutrients actually. This was how its structures looked when we had it in the feed, and this was how the structures looked when it was collected from the ileum. And again, you'll see here, this layer is actually still full of different nutrients, and looking on the digestibility, you'll see that it has a negative impact on the digestibility here in the bran relative to the flour. And I believe that if we had added, for instance, enzymes, we will have part of this digestibility moving up somewhere here. Now to my last point on the exogenous enzymes, because what I mentioned and what I started out to talk about, that is that the fibers, they represent some of the structures which cannot be handled by the endogenous enzymes, but we have the possibilities of adding exogenous enzymes, and some of the next presentation, they will address this to a larger extent, but it's also clearly so, and now you can get some of my view on what is possible. You can challenge me if you do not agree, but it's at least so that I have some, by looking on structure, I at least feel that there is possibilities by using exogenous enzymes, but there is also clearly some limitation, and at least I haven't been seeing that they are not true so far. Well, first of all, we can say that possibilities with exogenous enzymes, we reduce the molecular size of the soluble NSPs. I mean, that has been known for years. It reduces the viscosity. It also gives rise to higher feed intake because you have a less viscous environment in the gastrointestinal tract. It will probably also reduce the endogenous secretion, meaning that we will need less endogenous secretion really to handle the same amount of feed passing through the gastrointestinal tract. And then it's also so that it will open up some cell structures. That means that we can have a higher digestibility of protein and other nutrients. And here I again is addressing that particularly the aileron cells has the potential to be increased in terms of digestibility, because it holds up to several percentages of the protein and amino acids, and is very, very high quality. So why not take use of that? Whereas some of the other structures here are more difficult to handle. Limitations with the exogenous enzymes, well, they relate to some extent to some of the structures we have here, very much depending on how particularly the Arabinose Islands, they are organized, and the cross-linkages between the different changes of Arabinose Islands, the side groups, and so on, they will give some limitation in how the different enzymes, they can handle this structure. But it's also clearly so that when you're looking on what is required to handle a cell structure or a chemical structure as the Arabinose Islands, you'll see that it's not enough that you only have silonases. You also need some of the other enzymes present to be able to handle this type of cell wall structure. And this is particular when you come to more complex composed fiber particles. This is indeed very, very important. And then in addition, I would also like to see that when I'm looking into, and I'm now speaking you should view it from the point of a grain and see what is really the potential, even with exogenous enzymes, to handle in those structures. Well, in my view, it's naturally so that the cell walls that's present in the endosperms, they can be modified by exogenous enzymes, and in principle that has been known for years. That is why you also see when you're adding exogenous enzyme, you'll see modifications of Arabinose Islands and beta-glucans, and a great part of that comes from these cell tissues during that process. In addition, it's also so that I'll see that this part of the grain will be able to handle to a large extent. We'll not be able to degrade all the polymers that's present in this cell wall materials, because there's still some limitation in terms of the structure of these materials. But the advantage will be that whenever you open up this cell structure, the endogenous enzymes will also get access to some encapsulated nutrients that's present. If you then are looking on the outer parts here, I must say I don't think that exogenous enzymes really will be able to do that much with them. And I'm building this on also data we have from for instance, from pigs, where we have isolated the outer part of wheat and rye and giving it that to pigs, and then fed the animals, and then looking on how much of these cell structures that could be degraded. And for some part of it, it's actually so that it's very close to nothing that disappears during passage of the gastrointestinal tract of a pig. Sow may be slightly different, and so is in general age of the animal will play a role. This picture here will not do anything, or if I should go to the protein-rich material, I think we see about the same pictures that the polysaccharides that's present in the cotyledon and the germ will be able to handle. To a smaller extent, also on some of the polysaccharides in the hulls of non-lignified materials, whereas when you come to, for instance, the hull layers of rapeseed meal and of rapeseeds and also of sunflower, I don't think we really can handle them to any larger extent. Okay, well, this was my take-home message. I think I've used more than 45 minutes. But as I said, fibers cannot be digested by the endogenous enzymes. There is diverse composition between tissues, cells, and protein-rich materials. The functional properties relates to solubility and viscous properties, encapsulation, and also that endogenous enzyme or exogenous enzyme can be used to alleviate some of the negative effects of the fibers in relation to feed intake, digestibility, and nutrient utilization. And with these words, thank you very much for your attention, and I'm ready for questions.
Thank you very much, Knud Erik. I'm sure that this very interesting presentation on fiber will raise a lot of questions. So at least we will start with a question on the screen, but if you feel that you have burning questions, do not hesitate to raise your hands. The first one, is there a method to analyze the NSP in ingredients that can be easily applied in a feed mill lab?
Well, when you're looking into the analytical scheme for the methods I was presenting, you'll see that it was quite, it takes some time to do the analysis, and I don't think you can apply that in a feed mill. I should though say that at present we are in connection with the European products, we are working with looking on the relationship between the different fibres fractions and also the NIR, and see whether it's possible to build calibration so that you can get some rough estimates of the fibers composition in different raw materials. How easy it will be, I am not completely sure, but I can say we are in process with that purpose, and that's also with the intention to see will it be possible to deliver something that is? To handle at a feed mill or other places where you can be interesting.
Second question, what is the higher and lower limits as percentage of feed of fiber to be included in layer feed for good performance, with and without NSPs?
I think I'll hand that over to one of the next speakers, because as I mentioned, I have I've been publishing with many different species, but I have never really been. I don't think I have one single publication where I've been dealing with layers. But it's naturally so that I can calculate from the data I showed here before, which is a typical diet that can be that's on the market in Denmark. You will end up with a total fiber concentration at around slightly below 20%. Yeah. And that is about what you typically see in diets for pigs, actually. And although it can be difficult to make similarities, I can say that physically a pig can handle up to around 25% of dietary fiber in a diet without too much that growth rate and performance will go down. If a layer hens will be able to handle the same amount, I'm not completely sure. And if it's so that you add NSPases, you will probably be able to add slightly more. Yeah. Okay. A bit, I don't know if it's bit or slight. I'm a little careful.
Yeah. Another question, what are the effects of feed processing on the fiber structure and their potential degradation by enzyme, for instance?
Yeah, feed processing, I did not address this at this presentation, but when you come to it will influence to some extent also some of the physical properties. You will be able to, depending on how severe you are doing your processing, you will solubilize a slightly larger proportion of the cell wall carbohydrate or polysaccharides. But I don't think in itself that you will influence that much how much you actually are able to degrade. that are primarily controlled by the structure of the carbohydrates and also lignification and so on.
But the hard processing, like for instance, extrusion
and expansion, they will change a little bit, and they will naturally change also some of the behavior of the material in the gastrointestinal tract. But if you then are looking on for the entire gastrointestinal tract, I don't think that there is that big a difference actually in how much nutrient you can get out.
Okay. So you mentioned the different NSP content and structures between the different materials. Within one particular feedstuff, what is the link between the variation in the NSP structures and their potential digestibility and so on within a raw material?
One raw material?
Yeah.
Well, you can say that in if you take, for instance, wheat. What matters for all the, I can say in general, if you take cereals, that is the feed material I know is the best. For instance, if you have in total you have a higher proportion of the hull and husk layer or pericarp and hull layer relative to the endosperm, that will definitely have a negative impact on it. When you have a different, it also depends a little bit on where you're talking. If you're talking in the small intestine, what's handled there, the structure in itself can have an impact on partly viscosity, but also how the polysaccharides, they will behave in that gastrointestinal part. Whereas I don't think when you take the entire gastrointestinal tract that there is that much, because what I've seen in most of the data I've been analyzing, that is that what really matters, for instance, for Arabinose Islands, that is the Arabinose to xylose ratio. And the higher, the more difficult for it to be degraded.
Yeah, and you think that today there is a variation in the structure, not only between
there's some variation in the structure, but I don't think those which are on the market is that dramatic.
Yeah.
I don't think there's so big a difference in it. For many years, I was doing an experiment with barleys, barley varieties that was, you can say, ordinary feed barley varieties, and also barley varieties that was suitable for malting. And there we could see that with the barley varieties that was suitable for malting, they had a lower content of beta-glucans. It also had thinner hull layers, and it had a higher, that was tested in experiment with rats, and it also had a higher digestibility of energy.
Okay.
Yeah. So there you could see some difference in it. And that's because basically when they have been selecting the varieties which were suitable for malting, they are selecting for low viscosity, because viscosity is also a problem when they are producing beers, and also for thinner hulls. Mm-hmm. Exactly the same. And I must also say that when I was at the meeting last year it was in Canada where they were presenting the data about some new varieties they had with rapeseeds, and that was rapeseeds that had particularly the thinner hulls.
Okay.
Yeah.
Interesting.
And I could also say that back in time we were running as products on rapeseeds where we were looking for the size of the seed, and they are actually related. They are related to the total fiber content, because if you have bigger seeds, less fiber, because you have more germ relative to the outer parts. Okay. So they are.
Good. Two related questions, in fact. considering the functions of fiber you mentioned, is it important to seek means of degrading this undigestible fraction? And the last question was, should we degrade fully the fibers? So it's...
No, I don't think you should degrade it fully. I don't think so. Because if, and I don't know, dirty eggs, that is, as I can understand.
No, should we fully degrade the fibers or not? I think that was the two related
that's a very theoretical question, because I don't think you ever can come to a point where you fully degrade the fibers. Not the way we handle it here. Even when you do it on pilot plant, it's very difficult to degrade all the fibers. But I don't think you should degrade all the fibers, because you also need structures within the gastrointestinal tract. That is also involved in the water regulation and all the things, and how the digester materials that is looking and keep it holding water and so on.
Yeah, so it's a compromise.
Job. It is indeed
we
a
need
compromise.
Fibers.
And that's the same when you're asking, is it good with more or less soluble relatively to insoluble fibers? Well, you could say basically when you feed, you will always have a mixture of soluble and insoluble. But I also think you should have a balance, because you can, if you just are giving the animals, for instance, a very fermentable fibers, you will see that you also will create other problems. For instance, one example that was with peas. Many years back, when we were growing quite a lot of peas in Europe, and peas, they are quite well degraded. But what was the problem was that was causing very, very sticky feces, and that was not that was for pigs, but probably the same will be the case for broilers. They were causing this sticky feces, which just were, when you had to clean the slaughter out afterwards, it was very, very difficult to handle. So you should have a balance, for sure.
Another question: what is the optimal particle size, talking about feed processing, of fibers to stimulate a good function of the gastrointestinal tract and the gizzard in particular?
It shouldn't be too fine, I'm pretty much sure. Right. And in many cases, I must say, I asked one of my colleagues who has been working more with poultry, and asked her whether it was so that we were using whole wheat grains for broilers, but as I understand, that is not really the case. So I don't think it should be too fine, but that is not a clear answer, I know. But I'm not an expert in that area.
Maybe some can comment on the use of whole grain or partially broken grain, and because that has shown some quite good potential on digestibility. Any more comments on this point? Yeah.
Do you think that there's a sense, there's an advantage to put lignin in the diet from lignocellulose, from meal from the forest?
I don't think that. I mean, with the raw materials you have available, I must say I don't think there is that much rationale in that. It could be that you should have some special properties of them, because whenever you have a more concentrated fiber source, you will naturally have other properties than what is possible with the natural fibers. But I think in most cases you can also fulfill what is needed with the natural fiber. So in most cases, it's not a problem of having too little fibers, actually. It's probably more a question of having the optimum amount, I would say. And I don't think you really need, because then you say, well, why not go for other types of cereals? As for instance, you can say a cereal as oats, which has a lot of lignin and cellulose in the husk layer, and even barley. Oats, that is close to 30% of fibers. So that's a possibility.
Yeah, there's two also related questions. One is what is the maximum impact of digestibility improvement using an NSPase? And on the bottom one, you mentioned that by breaking down NSP, you release amino acids for digestion. Can you quantify? So in both cases, what is the maximum you can release with NSP degrading enzyme.
If I should take it from for instance, I think it's easiest to talk about, because otherwise I need to do several calculations. But if you take you take a wheat as an example, how much of a wheat can you degrade from a fiber perspective? You will have around 7-8% in the pericarp tester layer. I don't think you can handle that. Then you have 5-6% of alveolar cells, and there you can probably completely handle in the order of maybe half, less than half, or sorry, more than half of this layer. So you will end up being something like 10% you can't handle at all for wheat.
And if we think about corn, for instance, which is totally different structure.
Corn is a different structure, but basically if you take the ratio between pericarp and tester and alveolar cell, because these are the cell structures which are most difficult to handle. It may be slightly higher for corn than for wheat, for instance. It may be.
Okay.
And there will also be, even in, when I'm saying endosperm, we more or less can handle, there will still be some of the structures you can't do anything about.
The first question I think we can let for the next pages, because it's really dedicated to layers. There is one question, but in fiber structure, where is located chitin? Do you have chitin in vegetable plants, I would say?
You will have chitin in some, but I must say I haven't been looking that much into that. I don't think I can give a clear answer, but you will not have chitin, for instance, in cereals for sure.
Yeah. And finally, is there a common worldwide qualification of fibers today? Do we talk about the same things?
No. That's for sure. But that I can recommend reading my chapter in the book, and there you can be advised.
So
well, the thing is
that, for your book.
Well, excuse me?
No, I say we are waiting for your book.
No. But I can say in general, it's so that the methodologies that's used for fibers, that is the complicated one which I'm presenting here. What is very, very common when you come to analyzing feedstuffs, that is to use the NDF and ADF methods. And basically, you can say that with, I can try to go back.
Yep.
I don't know do it. But basically, what you have with the NDF, that is that they are relatively close to the measurements you have with the methods I showed on the screen here in terms of insoluble fibers. Mm-hmm. So, but that is a sum of cellulose and insoluble hemicellulose and lignin. And then you have the acid detergent fibers. That is reasonably close to cellulose and lignin. And then you can naturally also go for acid detergent lignins, which are somewhat to estimate of the lignin content. And in addition, it's a crude fiber, for instance, that is generally lower, and it's less than cellulose and lignin. So that's basically what you get. So with the same feedstuff, depending on how you analyze them, you can have data that varies by a factor of 3 to 6, depending on the matrix, on the same feed.
So be careful when comparing data from different tables or different books.
Yeah.
Maybe more questions from the audience before the coffee break? Feel free to ask questions. It's not only using Slido. So no more questions? Sure. So you can address more questions during the coffee break to Knud.